this is chapter 20, gram-negative bacilli of medical importance. Okay, so aerobic gram-negative non-enteric bacilli. It's a large, diverse group of non-spore-forming bacteria. It's a wide range of habitats. It lives in the large intestine, which are called enteric. Zoonotic, which means they transfer the infection from species, a different species to humans, from animals, different animal species to humans. Respiratory, soil, and water is where they all hang out or live. Most are not medically important. Some are true pathogens, though, and some are opportunistic. When we lose certain areas, like antibiotics, give rid of good bacteria, these take over. All have outer membrane liposaccharides of the cell wall, which produces our endotoxins. So endotoxins are from the lipopolysaccharides on the cell wall. Okay, Pseudomonas and Burkholderia are opportunistic pathogens. Brucella and Francisella are zoonotic pathogens transferred from different species. Bordetella and Legionella are mainly human pathogens. Okay. Gram-negative rods, okay, they're aerobes that do not ferment carbohydrates. Pseudomonas is one, Burkholderia, Bordetella, Francisella, and Alcagenes. Okay. Facilitative anaerobes that ferment carbohydrates. Okay. Lactose fermenting are Escherichia, Klebsiella, Citrobacter. Okay. Non-lactose fermenting, they're oxidase negative. Those are Salmonella, Shigella, Proteus, Providencia, Morganella, Hafnia, Edwardsiella, and Ceratea, and Yersinia. Okay. Oxidase positive gram-negative pathogens are Haemophilus and Pastorella. The obligate anaerobes that have to have with no oxygen are your Bacteroides. Okay. This is a flow chart or a chart of the different types of pathogenic gram-negative bacilli, and we'll go through each one of these. This is kind of a summary of what we're going to cover here. Okay, those are the two charts there, and this is your small the Pseudomonas. Okay, those are small gram-negative rods with a single polar flagellum. They're free living. They're found primarily in your soil and seawater. Okay, fresh water, and they also colonize plants and animals. They're important decomposers and bioremediators. Okay. They frequent contaminants in homes and clinical settings. They use aerobic respiration. They do not ferment carbohydrates. They produce oxidase and catalase, and many produce water-soluble pigments. The Pseudomonas aeruginosa, that was the first one in the chart. They're common inhabitants of soil and water. Intestinal residents, about 10% of normal people have these bacteria. They're resistant to soaps, to dyes, to quaternary ammonia, which is our disinfecting compounds, disinfectants, okay, drugs, and drying out. Frequent contaminated of ventilators in hospitals, IV solutions, and anesthesia equipment. That's where they're commonly found. They're opportunistic pathogens. They take over in spaces where they're given a chance to. That's what they look like, and this is the type of infections that occurs with the Pseudomonas aeruginosa. Okay, you see nasty types of infections. You can see the rod-shaped bacilli. Pseudomonas aeruginosa is a common cause of nosocomial infections, okay, in hosts with burns, neoplastic diseases, and cystic fibrosis. Remember, nosocomial infections are infections from you contact while you're in the hospital. Those are nosocomial infections. Complications of these infections include pneumonia, UTIs, abscesses, otitis, and corneal disease. So eyes, ears, and other types of abscesses in urinary tract. Endocarditis, meningitis, bronchial pneumonia. It's a grape-like odor, which is greenish bluish pigment, which is pyocyanin, and it's multi-drug resistant. Okay, it's kind of hard to treat those things. Okay, Pseudomonas aeruginosa, that's what they look like in the agar plate. And you see the ring around each one of the colonies. Okay. Related gram-negative aerobic rods are the genre Burkholderia, Actinobacter, Stentrophomonas, are similar to the Pseudomonas. They have a wide variety of habitats in soil, water, and related environments. They're obligate aerobes. They don't ferment sugars, but they're motile and they are oxidase positive. That was that ring around you saw on the Niagara Plains, that ring around the colonies. And they're opportunistic to take advantage of areas that have become available to it. Burkholderia cepacia is active in biodegradation, okay, a variety of substances. As an opportunistic agent in the respiratory tract, urinary tract, occasionally skin infections, and is very drug resistant. Burkholderia pseudomela, generally acquired through penetrating injuries or inhalation from environmental reserve where it's located. Wound infections, bronchitis, pneumonia, and septicemia are the cause of that bacteria. This is the genus Burkholderia. You can see the infections with nodules in the lungs here, kind of similar to tuberculosis bacteria. Okay, and the rod-shaped gram-negative bacteria. Streptophomonas maltophilia is a natural habitat. It appears to be fresh water and soil. It's associated with plant roots. Okay, it's also a member of the fecal microbiota in humans and may be isolated from domestic animals. In the clinical setting, primarily as in contaminated disinfectants, you have dialysis equipment, respiratory equipment, water dispensers, catheters. It forms this biofilm with it, which protects it against drugs and disinfectants. And that's what it's an opportunistic bacteria. That's where it lives. Infections occur in cancer patients, people with indwelling invasive dialysis. Okay, patients have those are easy to get that infection of the Streptophomonas. Okay, one of the most drug resistant bacteria that we have. Okay, very resistant to almost all antibiotics. Therapy with a combination. Okay, when we have doing with bacteria that are very resistant to antibiotics, we usually use combination therapy. Combinations of trimethoprim and sulfamethoxylates are usually effective. So the main reservoir for Pseudomonas aeruginosa is what? Soil and water, infected individuals, asymptomatic carriers, or wild animals. Where does Pseudomonas aeruginosa?
Soil and water.
Soil and water. That's where it lives. You got it. Perfect. Okay. Brucella and brucellosis. These are tiny gram-negative coccobacilli. So very small ones. There's two species. Brucella abortus, which is found in cattle, and Brucella suis, which is found in pigs. Brucellosis is also called malta fever. It's also called unland fever, and it's also called Bain disease. All those are brucellosis. It's a zoonosis transmitted to humans from infected animals. If it's arbotorus, it's transmitted from cattle. If it's cellular, it's transmitted from pigs, and it causes brucellosis. It's a fluctuating pattern of fevers, which can last several weeks up to a year. Combination of tetracycline and rifampin or streptomycin is used to treat these bacteria. The animal vaccine there is an available animal vaccine for it, and it is a potential bioweapon because it can transfer to humans from other species. It makes a very potential bioweapon for it. Francisella tularensis, okay, causes tularemia, which is another zoonotic disease. Okay, this is a disease of mammals in the northern hemisphere, particularly rabbits. That's tularemia. Okay, transmitted by contact with infected animals, infected water or dust, or bites by vectors like fleas, mosquitoes, or ticks. Causes headaches, backaches, fevers, chills, malaise, and weakness. Okay. It also causes ulcerative skin lesions, swollen lymph glands, conjunctive inflammation, which is around the eyes, sore throat, and intestinal disruption. 10% death rate in the systemic, where it enters the body, and pulmonary forms. Okay, so when it enters into the bloodstream or enters into the lungs, you got a 10% death rate associated with Francisella tularensis. Okay, it's treatable with gentamicin and streptomycin combination. Gentamicin is a very powerful antibiotic that we have. A lot of times, gentamicin causes deafness or inner ear problems, problems with your equilibrium, problems with balance. So we don't like to use that if we can, but in this case, we have to use it. It's such a nasty bacteria. Intercellular persistence can lead to a relapse after treatment. Okay. No vaccine is currently available for this disease. Often considered one of the most infectious of all bacteria. Again, because it's so infectious and because it's so hard to treat, it is a very potential bioterrorism agent. Bordetella pertussis. Okay, it's minute, very small. It's encapsulated coccobacillus, causes pertussis, or what's called whooping cough. Okay, communicable childhood affliction. It's an acute respiratory syndrome. It's often severe and life-threatening complications in babies. Okay, reservoir is in apparently healthy carriers. So healthy adults usually carry it. Transmission by direct contact or inhalation of the aerosols. And you can see the expansion of the lungs. You see even the rib cage here as they're trying to expand to take breathers. They're coughing from the whooping cough, pertussis. The virulence factors is receptors that recognize and bind to ciliated respiratory epithelial cells. So it binds to the ciliation of the respiratory cells in the bronchioles, in the bronchial tubules. Toxins that destroy and dislodge the ciliated cells. It breaks up and destroys that ciliated lining. So you cough and you can't bring things up. That's, you have that hacking cough. So you get loss of the ciliary mechanism, leads to buildup of mucus, and then mucus blocks the airways. You get that very forceful concentrate. You even can't get down there because the cilia has been destroyed. Vaccine is the DTaP vaccine. It's an acellular vaccine with toxoid and other antigens on it. This is Bordetella pertussis, how it's been reported in the years. You see spikes in 2005, 2015. Okay, the genus Alcogenes lives primarily in soil and water. Okay, that's where it's found. May become normal flora in the human body. Alcogenes fecalis is the most common clinical species. It's isolated from feces, from sputum, and from urine. Occasionally associated with opportunistic infections, causing pneumonia, septicemia, and meningitis. Legionella pneumophilia, okay, widely distributed in water, closely associated with amoebas. Okay. This causes Legionnaire's disease. Outbreak of pneumonia in 1976 afflicted 200 American Legion members and killed 29. Legionnaire's disease, okay, the most severe is what's called Pontiac fever, prevalent in males over 50. Nosocomial disease in elderly patients, that's what they get in hospitals. Drinking fountains, things like that can be easily transmitted, nosocomial infections in hospitals to the elderly. Not contagious otherwise. I mean, patient to patient, it has to be through a water supply, usually with amoebas. Fever, you get fever, cough, diarrhea, abdominal pain. Pneumonia fatality rate is between 3 to 30%, which is a big variance size because of it affects the elderly. You get large range like that. Okay, so 3 to 30% only in Legionnaire's disease. Treatment is levofloxacin and azithromycin. Azithromycin is the same treatment, one of the drugs used for AIDS treatment also. This is what it looks like on the microscope. You can see the colonies here. Okay. And this is the bacteria. This is the amoeba cell. And amoeba cell hosts the bacteria. So family, Enterobacteriaceae, are gram-negative bacteria. It's a large family of small, non-spore-forming rods. Many members inhabit soil and water, decaying matter, common occupants of large bowels of animals. Most frequent cause of diarrhea is through enterotoxins. Now, endotoxins are on the cell membranes. Enterotoxins are secreted, okay, and they target the digestive tract, the intestines, causing diarrhea. So when you see the two terms, we'll see the two terms in nursing exams. And endotoxins are on the cell wall of the bacteria. Okay, enterotoxins are secreted and specifically attack the intestines. So enterics, along with Pseudomonas, account for almost 50% of the nosocomial infections acquired in a hospital setting. The most important enteric opportunists are E. coli, Klebsiella, Enterobacter, Serratia, Proteus, and Citrobacter. Okay, the family Enterobacteriaceae are glucose fermenting, nitrate reducing, oxidative negative. Okay, most have a flagella present, and they're usually regular straight rods. There's two types. There's coliforms and non-coliforms. Coliforms ferment lactose. Non-coliforms do not ferment lactose, do not break it down. Enrichment, selected and differential media are utilized for screening samples for the pathogens. So we can use different media to see which of the pathogens that you have. Okay, this is lactose negative. This is lactose positive with the rings. Again, lactose negative, lactose positive with the rings around it. Lactose positive, lactose negative. These are biochemical traits separating each one. The sonoid, you have a bacteria, and we're going to start whether it's lactose negative, lactose positive. From there, it has motility. From there, phenylalanine is one of the reduces the nitric acid with phenylalanine. And we'll go through each one of those steps here. This is the overview of the flow chart of it. And this is the test that we use. So this is for isolating, identify, selected, enteric genre. Okay. The first one is a Mecongri agar containing biosalts and crystal violet to inhibit gram-positive bacteria. That's where you separate out the gram-negative bacteria. Contains lactose and neutral red dye to indicate pH changes and differentiate the rapid lactose fermenting bacteria from non-lactose fermented. So the first step here was lactose fermenting or non-lactose fermenting. Okay. Lactose positive colonies are red to pink from the accumulation of acid acting on the neutral red indicator. Okay. The next step is the Levin's EMB agar. Contains biosalts plus eosin. Eosin is a dye and methylene blue dyes, okay, that cause lactose fermenters to develop a dark nucleus and sometimes a metallic sheen over the, over the surface. Non-lactose fermenters are pale lavender and are non-nucleated. Then hectoin enteric agar was the next step. Also contains biosalts and especially good for isolating pathogens. We use bromophenol blue and acid diffusion indicators to differentiate lactose fermenters, salmon pink to orange from the non-lactose fermenters. Green, blue, green, and blue-green are also detected the production of sulfuric hydrogen sulfide gas, which in turn turns the colony a black color. H2S is sulfuric acid. This is the one that is put into, if you have a gas leak in your home, it's natural gas. Natural gas has no odor. We inject hydrogen sulfide, so hydrogen sulfide gas, which causes that smell, that rotten egg smell. That way, if you have a gas leak, you can smell it, and you can do the appropriate action. Utilization of lactose as a disaccharide. Lactose is a disaccharide, requires two enzymes: a penase to bring lactose into the cell, and beta-galactosidase to split it into monosaccharides, glucose and galactose. Okay, rapid fermenters have both enzymes, so it can bring in the cell and then break it apart. Okay, ONPG is chemical shorthand for the test that detects these slow lactose fermenters. Okay, these are all on that slide, all on that flow chart. TSI is a non-selective media that indicates some combination of fermentation reactions, primarily lactose and glucose, and possibly sucrose, by means of what's called phenol red, which is an indicator dye. It also reveals gas and hydrogen sulfide gas. Okay, hydrogen sulfide gas production. Hydrogen sulfide gas is a product reduction of sulfur and is indicated by reacting with iron salts to form the black precipitate of ferric sulfide. That gives it that rotten egg smell. The indole test tests the capacity of an isolate to cleave a compound called indole off the amino acid tryptophan. Tryptophan is one of the 20 amino acids. It's most common in turkey, and tryptophan is the main ingredient in melatonin, which explains why after Thanksgiving coming up, which is going to be in a week or so, you eat lots of turkey. What do you do after make Thanksgiving dinner? Go take a nap, right? Everybody gets tired. Okay, that's because of tryptophan. This is a precursor to melatonin. So turkey has a lot of tryptophan in it, tryptophan melatonin. Melatonin makes you tired, and you get sleepy after a big turkey meal. That is because of the tryptophan amino acid that's high prevalence inside of turkey. And the tryptophan, the cleaving occurs. Covector agent reacts with indole to form a bright red ring at the surface of the tube. The methyl red test indicates a form of glucose fermentation, in which a large amount of mixed acids accumulate in the medium. The pH lowers around 4.2, which is very acidic. Normally around 7 is neutral. The lower the pH, the higher the acid, stronger the acid. So the methyl red dye remains red when added to the tube. Voges Prosacker VP test determines whether the product of glucose fermentation is a neutral metabolite called acetyl methylcarbinol, acetone. The subject reacts with Barrett's reagent to form a rosy red tinge in the media. The citrate media contains citrate as the only usable carbon source and a pH indicator. Prothymol blue is an isolate, can utilize this carbon source. It produces alkaline byproducts. Okay, so the opposite of the acid, these are alkaline, these are basic byproducts. Raises your pH, results in converts the medium's color from green neutral to blue, which is alkaline. Again, the pH goes up greater than 7. Lysine and ornithine decarboxylase, LDC and ODC, detect the presence of enzymes that remove carbon dioxide from the amino acids, the end product of alkaline amino acids that raise the pH. Again, raising pH is basic, okay, cause the pH to increase. pH decrease, it's acidic. The urea media contains 2% urea, which is a urine byproduct. It's a nitrogenous waste product of mammals. Phenol red is your pH indicator. The enzyme urease hydrolyzes urea into ammonia, which raises the pH. Okay, ammonia is a base, raises your pH, and turns the indicator bright pink. Phenylalanine. Phenylalanine deaminase is an enzyme produced primarily by the protease group. So if it breaks down phenylalanine, it's another one of our amino acids, okay, produces phenylpyruvic acid. Okay, that means it is a protease bacteria. This reaction is visible with ferric chloride. Okay, it turns olive green when it reacts with the phenylpyruvic acid. Motility is tested by hanging the drop slide and seeing how mobile the bacteria is. Okay, these are different opportunities to Enterix. This is different testing of it. This is your indole, methyl red, and this did the different tests, whether they're going to be positive or negative as it goes through. These are different species. All right, so the antigenic structure, the virulence factor, how virulent are these bacteria? They're complex surface antigens, what we use to identify the bacteria when our immune system recognizes it, contributes to the pathogenicity and triggers the immune response. Okay, those are your surface antigens. H is the flagellar antigen on the flagellum. K is the capsule or fimbrial antigen, and O is the somatic or cell wall antigen. So not all species have capsules and flagellum, but they all have a cell wall. So not all species carry the H and K antigen, but all have the O antigen. We all have a cell wall. Okay? These are implicated in endotoxic shock syndrome. The pathogenesis of enterics is also tied to the production of endotoxins or exotoxins. Remember, endotoxins are on the cell wall. Trigger the response, you get your conditions of your disease from that. Exotoxins are secreted and directly attack the intestines. Here's your antigens. This is somatic O antigen, which is on your cell wall, which is present on all bacteria. The flagella H antigen and the capsule K antigen are only present on some. Some have capsules, some don't. Some have flagella, some don't. But all have O. Which of the following is true of enterics? Some are normal flora, some release endotoxins, some release enterotoxins, or all of the above.
All of the above?
All of the above, yep. E. coli is the most prevalent enteric bacillus. Most commonly aerobic and non-fastidious bacteria in the gut. There's 150 strains of E. coli. Some are virulent with plasmid transfers, a little ring of DNA that transfers. Others are opportunistic. The pathogenic strains of E. coli, the pathotypes, can be grouped as enterohemorrhagic, which is E. coli EHEC, which is hemorrhagic syndrome and kidney disease. Okay. Enterotoxigenic, which is severe diarrhea, okay, due to heat labile toxin, heat stable toxin, stimulates secretions and fluid loss, also has fimbric with it. Okay. So E. coli, like I said, it's the most prevalent and is enteroinvasive, okay, inflammatory disease of the large intestine. Enteropathogenic, okay, infantile diarrhea, wasting from that. Enteroaggregative, common cause of pediatric diarrhea, and diffusely adherent E. coli, which is a minor cause of pediatric diarrhea also. All of them affecting the intestine. So most clinical cases are treated exclusively among humans. Pathogenic change frequent agents of infantile diarrhea. The greatest cause of mortality among babies is E. coli. Causes about 70% of travelers' diarrhea, When you go to different countries, you get the water, different bacteria in it. They attack your gut, and that's traveler's diarrhea. Causes 50 to 80% of UTIs. Oral antimicrobes may be effective in the early phase of infection. One of the drugs we use to treat UTIs is Keflex, which are cephalosporin. We find in the early stages, cephalosporin is used, and Septra are also used in treating it. Keflex is the new drug that we use to treat it now for most cases of UTIs. We get it early enough, it treats it very well. Over-the-counter preparation for symptom relief, kaolin or Imodium. This slows your gut from moving from motility, so it can severe to retain the pathogen longer than the body fight it. Okay. Bismuth salicylate mixture, which is Pepto-Bismol, is more effective actually. It counteracts the enterotoxin and also provides an antimicrobic effect. Okay. The E. coli in food is O157H7. It's named for the different, and the O antigen and the H antigen. Okay. It's an enterohemorrhagic strain involved in infection from fast food hamburgers in 1982. The reservoir was in a cattle intestine, is where it was found, and transferred into it was a zoonotic infection, transferred into the human population. And it was from fast food hamburgers. Virulence due to a cell wall receptor that can fuse with the host cell membrane, creates a direct port, okay, into which the bacteria secrete their toxins. Toxins enter the host cell, bind ribosomes, and disrupts protein synthesis, leading to death and shedding of intestinal cells. So currently testing in commercial meat is performed randomly, so we check it. The USDA, United States Department of Agriculture, requires that all fresh meat and poultry products bear warning labels to remind consumers of safer handling. Other coliform infections. Okay, most of these can affect nearly any organ. They're opportunistic, and a lot of them are drug resistant. They take more than one type of antibiotic to fight the infection. Okay. Klebsiella pneumoniae, normal inhabitant of the respiratory tract, has a large capsule, causes nosocomial pneumonia. Okay, meningitis, bacteremia, okay, wound infections, and UTIs. Enterobacter, okay, causes UTIs and from surgical wounds. Again, nosocomial infection. Citrobacteria, opportunistic UTIs and bacteremia. Serratia marcescens produces a red pigment, causes pneumonia, burn and wound infections in the hospital, septicemia, and meningitis. Okay. These are the coliforms you see on the different colonies. Non-coliform, lactose negative enterics. Opportunistic is a genre. You have Proteus, you have Morganella, and you have Provincia. True enteric pathogens are Salmonella and Shigella. Okay. Opportunists like Proteus, and ones that are similarly related to it, they're ordinarily harmless sapros, which means they can sort of live within oxygen, found in soil, manure, sewage, polluted water, and commensals of humans and animals. The Proteus is a swarm of surface and moist agar in concentric patterns and are involved in UTIs, wound infections, pneumonia, septicemia, and in infant diarrhea. The Morganella morgani, okay, and Providencia are involved in similar infections. And you see again the different colonies of the Proteus and the bacteria like it. Okay, the Salmonella and the Shigella, well-developed virulence factors, primarily pathogens. They're not normal human flora. Okay, Salmonella and Shigella are not in your normal human flora. Salmonellosis and Shigellosis, some of gastrointestinal involvement and diarrhea would often affect other systems too. The genus Salmonella contains two species, Salmonella bongori and Salmonella enterica. The Salmonella bongori is found only exclusive in cold-blooded animals, so reptiles. So if you're handling reptiles or handling pet reptiles, the Salmonella bongori can transfer as a zoonotic infection to the human species. That's bongori. The enteric has 6 different subspecies. Each belongs to one of over 2,500 serotypes, reflecting the source of where it comes from. It comes from many different sources. But the bongori one comes from cold-blooded animals. The formal designation of Salmonella serotype is its genus subspecies name followed by the serotype and the serotype name. Okay, often abbreviated with genus and serotype. So flagellated, survival outside the host, resistant to chemicals, biles, and dyes. The prevalence of salmonellosis, you can see spiked in 1986. You can see a constant trend. Okay. Typhoid fever. Typhoid is caused by Salmonella serotype typhi. Bacillus enters with ingestion of food, of water contaminated by the feces, some by close personal contact. Asymptomatic carriers, some chronic carriers, shed the bacilli from the gallbladder. Bacilli adhere to the small intestine, cause invasive diarrhea that leads to septicemia. Treatment is with ciprofloxacin and cetrotriaxone. Okay, so 2 antibiotics. And 2 vaccines are available for temporary protection of typhoid. Typhoid fever, this is a typhoid bacilli. Get into the what are those called? I'm drawing a blank on the pili of the small intestine. Line the wall, they get into there and go into the bloodstream, causing septicemia. They're carried to the liver or the spleen by the blood vessels. You see the intestinal wall, and it infects the intestinal wall. And you see the perforations in the intestinal wall called the ulcerations. And you see some of the ulcerations inside the intestinal wall right there from the typhoid bacilli. Shigellosis is a common incapacitating dysentery disease. Fever, nausea, painful abdominal cramps, frequent defecation of watery stool filled with mucus and blood. Etiology agents are S. dysentery, S. soni, S. flexneri, S. boydii. Invades the villus of the large intestine, does not perforate intestines or invade the blood. So not like the typhoid one, which it does perforate the intestines and invades in the blood. The shigellosis does not. Okay. It invades the villus of the large intestine. So the extensions into the large intestine that absorbs waters and salt from the large intestine. It enters Pierre's patches. It instigates inflammatory response of your endotoxins and exotoxins. Okay. Again, endotoxins are produced inside the body, like inside the bloodstream, inside the intestines. Exotoxins are produced outside the body. Okay, like onto the surface of the skin and things like that. Treatment is fluid replacement and ciprofloxacin and sulfotrimethoprim. All bacteria, you'll notice nursing, all bacteria is involved with all diarrhea. The main treatment is fluid replacement. The main cause of death in diarrhea is dehydration because water just comes out and water body can't retain it. So it's fluid replacement, either IV or just constant fluid with electrolytes. Shigella and bacterial dysentery, you can see what it does to the intestine. You can see how it infects the different areas of the intestine, all the different villi there. Okay. So if your nephew gets a gastroenteritis infection of diarrhea after cleaning out his pet snake's aquarium, okay, the most likely pathogen is what? Shigella dysentery. What's that?
Is it Bangori?
That's Bangori, because it is
cold-blooded.
Cold-blooded. And is a zoonotic infection which is transferred to human. So it's Salmonella Bangori. Okay, very good. Enteric Yersinia pathogens. Yersinia enterocolitica, domestic and wild animals, fish, fruits, vegetables, and water. Bacteria enter the small intestine mucosa. Some enter the lymphatic system and survive in phagocytosis by getting inside the cell. Inflammation of the ileum can mimic appendicitis. Okay, because remember the appendix is the small intestine, you have the duodenum, jejunum, and the ileum. So you have your small intestine wraps around and then feeds into the large intestine. So this is your small intestine, and then it's got the sac. Remember, this is the duodenum. This is the jejunum. And this is the ileum. Okay, the ileum feeds into the cecum right here, which is the beginning of the large intestine. And right behind the cecum, this little finger-like distance here, that's your appendix. Okay, so inflammation of the ileum. If this gets inflamed here, this can mimic the signs of appendicitis because it's right where the appendix is. So Yersinia enterocolitica can, when it gets infected, because it infects the ileum of the small intestine, can mimic appendicitis. Yersinia pseudotuberculosis, infection similar to Y enterocolitica, but more lymph nodes are infected with this one. The pestis and the plague, the bubonic plague, non-enterotic bacteria. They're tiny gram-negative rods, unusual bipolar staining, and capsules. Virulence factor. They're capsular and envelope proteins to protect against phagocytosis and foster intracellular growth. So they protect it and they grow. That's what makes the plague so devastating. The coagulase produce an endotoxin and a murine toxin. Okay, this is Yersinia pestis and the plague. And see what the bacteria, the nuclei look like here. Okay, so humans develop the plague through contact with wild animals, the Salvatic plague, domestic or semi-domestic animals, okay, or infected humans. It's found in 200 species of mammals, rodents, without causing the disease. So rats, mice, hamsters, guinea pigs, they can all carry the plague, okay, without being sick themselves. Flea vectors, bacteria replicates in the gut. Coagulase causes blood clotting that blocks the esophagus, and fleas become ravenous and feed on those areas. So this is the usually what do we got here? The flea, the chipmunks, and through aerosols or through another flea biting an affected squirrel, okay, can feed into the humans by droplets, and it's from the bubonic plague and the pneumonic plague. So droplets become the pneumonic plague. Infections by the flea, if it bites you, can be the bubonic plague. So it's mostly animal to human infection. The infection dosage is between 3 to 50 bacilli, so not a lot. The manifestation infection leads to the bubonic plague, to septicemic plague or pneumonic plague, depending on how it's introduced in the body. The bubonic plague, this bacillus multiplies in a flea bite, enters your lymphatic system, causing necrosis and swelling of lymph nodes, called a buba. A buba is a swelling of your lymph nodes. You could use it in your groin or your axilla, which is high concentration of lymph nodes. Septicemia plague, progression to massive bacterial growth. Virulence factor causes intravascular coagulation, subcutaneous hemorrhage, and purpura, which is the black plague. Pneumonic plague, localized to the lungs, highly contagious because it's droplets from aerosols now, and it is fatal without treatment. Okay, so those are 3 types of the plague. Bubonic, where it swells the lymph nodes, the bubo was the swollen lymph nodes. Septicemia, where it's intravascular, and pneumonic, which in the lungs, which is that's the one part that's transferred from human to human because it's vaporized in your droplets, and it's fatal without treatment. Diagnosis, treatment, and prevention. Diagnosis depends on history, symptoms, and lab findings. From aspiration of bubbles, okay, you aspirate the lymph nodes. Treatment, streptomycin, tetracycline, or chloramphenicol. Killed or attenuated vaccines are available. We have vaccines for the plague. Prevention by quarantine, control of your rodent population in human habitat. Where humans are, we control our rodent population. Okay, oxidative positive, non-genetic pathogens in family Pasteurella. Pasteurella is a zoonotic genus. Okay? Again, spread from animals to humans that occurs as normal microbiota in animals and is mainly a concern to veterinarian. Okay, 6 recognized species among them: Pasteurella multicida, broadest spectrum opportunistic, Haemophilus influenza, Haemophilus influenza subtype, Agriophytus, H. Neuseri, H. Parainfluenza, H. Afrophilus. Okay, the Pasteurella multicidia is a zoonotic genus, normal flora in animals. It's an opportunistic infection, and many hosts are domestic animals with relatively close human contact. Animal bites, scratches cause local abscesses that can spread to joints, bones, and lymphatic nodes in humans. Immunocompromised are at risk for septicemia because they can't fight the infection and complications from that. Penicillin and tetracycline are your two types of treatments. Blood-loving bacilli. Haemophilus, tiny gram-negative pleomorphic rods. They're fastidious, sensitive to drying, temperature extremes, and disinfectants. None can grow on blood agar without special techniques called chocolate agar. It requires Hemin, NAD, and NADP. Some species are normal colonists of upper respiratory tract or in the vagina. Okay. Others are virulent species responsible for childhood meningitis and cancroid disease. H. influenza is acute bacteria, can cause acute bacterial meningitis, epiglottitis, otitis media, sinusitis, pneumonia, and bronchitis. Subunit vaccines called HIB is used in children in combination with Tdap for Haemophilus meningitis. This is what Haemophilus looks like. You can see the fluorescent glowing of it on the stain. And you can see that the Hib vaccine was introduced and it went from number 10. This is a million cases, 10 times 100,000. And that was in 1990. And by 1994, you can see how fast it's dropped. And it's stayed down ever since, once we have the vaccine. Haemophilus influenza, subtype agaphitis, primary agent of communicable conjunctivitis. Communicable conjunctivitis pink eye. It's very contagious. If your child has it, they send you home from school because it is a communicable disease. Haemophilus duceria is an agent of cancroid, which is a sexually transmitted disease. And parainfluenza and aprophilus are members of normal oral and nasopharyngeal flora involved in infective endocarditis, where it can lodge on the valves of the heart and cause damage. And this is pink eye. Pink eye is inflammation of the conjunctiva around the eye. And pink eye is very painful. The bubo of the bubonic plague is a ulcer where flea bite has occurred, granuloma in the skin, enlarged swollen lymph node, or infected sebaceous gland. Which one is the bubo?
I think it's A. I'm not sure.
I remember the bubo was your lymph node. Let's see where we had it. No, I had it here somewhere. Yeah, buboes. Aspiration of the buboes. There we go. Bubonic plague. Bubonic plague, most flea bites enter the lymph, causing necrosis and swelling of the lymph nodes. Swelling of lymph nodes is called a bubo, usually in the groin or axilla. So the bubo in the bubonic plague refers to enlarged swollen lymph nodes. Now it's transmitted by the flea bite. So it's an ulcer where the flea bite occurred, but that's not a bubo. That's the infection site. The actual term bubo is a swollen lymph node.
Oh, okay, got it.
Make sense?
Yes.
Perfect. Swollen lymph node.